Introduce our first speaker today, Oscar Vargas, who will be presenting on the phylogenetics of high Andean wood daisies.
Okay, thank you so much, Erika, for the introduction. I'm super excited. Can everyone hear me well? Can you hear me? Okay, great. So yeah, I'm super excited to talk about my research on Diplostephium and Linochilus. For those of you that know my work, I've been studying Diplostephium for more than 20 years since I was an undergraduate. So I tried to condense a lot of the highlights of my research of the last 20 years in this topic today. So, when I was an undergraduate, I wanted to study a genus of plants in the Astereae. I have always loved Astereae. And I wanted to do some sort of like treatment, monographic revision, maybe a key. So my advisor at the time suggested that I study Diplostephium. This was in 2004, more or less, and around that period, we actually didn't know how many species were in Diplostephium, but we know that there were like more than 100 species. And for those of you that have not seen these plants, this is how they look. So they are woody daisies. They are in the tribe Astereae. They live primarily in high Andean ecosystems. So in the northern Andes, they're going to live in the Paramos, in the central Andes, in the Punas, and they also make part of the high Andean forest. So you can see here is a picture of me when I was doing fieldwork in Colombia. This is one of these species that is a forest species. So it's woody, pretty big, big leaves, really nice. When you go up in elevation, then you pass the tree line, then you're gonna have more morphologies like these morphologies that are here at the top. So they are gonna be more shrubby. The leaves are gonna be smaller. Um, in relation with the capitula, when the species that are in the forest are typically going to have a inflorescence with multiple capitula, like you can see here in D. So these flowers belong to A. Then you go up in elevation, you're going to see a reduction on the number of heads. And then once you get really high in elevation, then you might have a reduction to having a single head. And many times when you have a single head, then you're going to have more flowers in those heads, which is kind of cool. So this is a map showing the distribution of the types of the genus Diplostephium before it was split, and I'm going to talk a lot about that in a second. But yeah, they're found in the cloud forest, as I mentioned, in the Paramo. So here, cloud forest and Paramo belong to the northern Andes, which is represented here in green. So mostly Ecuador, Colombia, Venezuela. There's also a little bit of Paramo in both Panama and Costa Rica. And then once you get to Peru, Bolivia, and Chile, which are the central Andes, you're gonna see that this area is a little bit drier. So when you get to the above the tree line, you are gonna see landscapes that are way more open, like the dry Puna, the desert. But I will say that most species of Diplostephium that you find in this area are gonna be in these slopes of the Andes that are facing the Amazon basin. So they are basically receiving all this precipitation from the Amazon. So, for my undergraduate, I wrote a key for the Diplostephium of Colombia, now mostly in Linochilus. Then in my master's, I did a Sanger sequence in phylogeny of these plants, mostly focusing on Colombia. And then for my PhD, I decided to do next generation sequencing and really expand the sampling to all South America, right? So for my PhD, I did a lot of field work in Peru and Ecuador, just trying to find more samples for my phylogeny. Obviously, one of the questions of the phylogeny was, well, is this genus monophyletic, right? So for that, I also sampled other genera that were hypothesized to be closely related with the genus. So this slide is pretty busy. These are pretty much the main figures of the paper that resulted from the first chapter of my dissertation. And basically, I use two types of sequencing. I use genome scheming, in which I assemble the entire cistron for the nuclear ribosomal region of DNA. So this contains both ITS and ETS. I assemble the chloroplast genome for all these samples and partially the mitochondrial genome. If you have worked with mitochondrial genomes, they're really complicated because they evolve too quick. The genes don't evolve, but the places of the genes are changing all the time. And I also use nuclear DDRAD um, markers for this. So these are basically going to be SNPs from the genome. And I don't have the number here, but it's in the thousands, like around 2,000, 10,000 SNPs. Okay, so let's look into the trees. So when we look at the trees, we can see that the nuclear DDRAD and the nuclear ribosomal are very similar trees to each other. We basically see one signal, and the signal is that Diplostephium, as considered before 2017, is non-monophyletic, is basically biphyletic. So we have two main groups, and there is a couple of exceptions here, but I'm going to talk about those in a sec. And we have a main group, a main clade that I call here A, which is in green. That makes a lot of sense with biogeography. This is the group that is mostly found in the northern Andes, so Colombia, Venezuela, and Ecuador. And then we have this other clade, clade B, that is mostly found in the central Andes, but it also reaches to Ecuador. So these two clades actually overlap in Ecuador and a little bit in Colombia. So you might be wondering, well, um, speaking of those two trees, well, what's happening with Parastrepia that is embedded there in one of the phylogenies, in the nuclear DDRAD, and in the nuclear ribosomal phylogeny, we see that there are two species of Diplostephium that are out of Diplostephium. Well, it turns out that there is a lot of hybridization in Diplostephium, specifically in clade B. And I'm going to show you a test that I did for this, but basically, hybridization is also what is biasing the position of certain species here. In this specific case, so Diplostephium cinereum and another species of Diplostephium that it has not been described, um, hybridized with Parastrepia. So that's why Parastrepia is embedded here, and that's why here these two species are sort of out um, because of that. So your next question might be, well, what's happening with the chloroplasts and the mitochondrial phylogenies? Well, when you look at those phylogenies sort of make no sense at all, biogeographically or morphologically, at least, what I try to do when I was trying to understand this. And what I came to the conclusion was is that there is so much hybridization here that is recent, and also there was ancient hybridization that we cannot really believe the chloroplast and the mitochondrial phylogeny anymore because I'm going to talk about this one because when we have hybridization, we know that the mitochondrial and the chloroplast genome are inherited only from one of the parents. So the hybrid is just going to have one of the signals for these genomes, right? What that means is that it's really the nuclear genome that receives from genetic information from both parents that is going to have that signal of hybridization and a better signal to infer a species tree. And this is something that I describe in the paper a lot, and I basically say, these chloroplast and mitochondrial phylogenies are basically gene phylogenies, which in this case, because of hybridization, we shouldn't trust as the history of speciation. And this is not something new. This is something that was proposed before by Risberg and Soltis in 91, in which they say, hey, chloroplast phylogenies are great, but we have to be really careful about using this because if there is hybridization, they are going to be biased. So I tested for hybridization with the DDRAD. Well, I actually tested in two ways. I'm just going to talk about the DDRAD here, but I use the ABA-BABA test. So the ABA-BABA test, what it does is it's going to look at SNPs in which the character states are incongruent with the phylogeny, like they are here. And basically, these patterns, these character states are incongruent for the phylogeny, and you expect these patterns to be equally distributed in your trees. Just so you expect 50% of Aba Baba and 60% of Aba and 60% of Baba in your phylogenies using SNP. So basically, you test for taxon, and then you can do this test and see that if there is significant difference between these patterns in your phylogenies. So this is the table from the paper, and this is showing that basically I have evidence for introgression or ancient hybridization among genera, so we call that ancient hybridization. I have introgression of hybridization inside Diplostephium group A, which is now renamed Linochilus, and also introgression inside Diplostephium group B. If you notice, here there is Diplostephium, this new species is the one that was placed in a different place, or let's say misplaced in the phylogeny, in the nuclear phylogeny with Diplostephium cinereum. So from this paper, I could say that there was a lot of hybridization, and I can say that Diplostephium is non-monophyletic, and it warrants basically the segregation of this clade into a different genus. So the type of species falls in B, so we need to rename A. So a year later, after I published the phylogeny, I did this. So I reinstated the genus Pyophontia, which was a name that was available for clade A. It turns out that it was a mistake, and it was a succulent mistake from my part, and I created 63 synonyms. The scientist, the botanist who realized my mistake was Patricio, and Patricio wrote to me and he said, hey, there is an older name that is available, and that's the one that should be used. So then we published another paper rectifying this mistake. Okay. I also want to say that this biphyletic nature of Diplostephium is being confirmed right now. So Gary Allen, who is here, is leading an effort to do a phylogeny of the Astereae. So I submitted some of my samples there. There's a preliminary phylogeny of this, and we can see where Linochilus shows in the phylogeny and where Diplostephium shows in the phylogeny. They're actually relatively far away from each other. And Linochilus is sort of in this grade with genera that are all over the plate, but mostly southern hemisphere. And Diplostephium sensu stricto is basically nested in this clay that primarily has genera from the Americas. So this is exciting to see that this is true, and we can see it in bigger phylogenies too. And one thing that I really wanted to show in this talk was the convergence evolution that happened in these two genera, right? So they're very similar to each other. They're difficult to differentiate. And there are some specific cases in which there are species that vegetatively are very similar to each other. So here in the left, I have two species of Linochilus. Here in the right, I have two species of Diplostephium, sensu stricto. And when we look at Linochilus rosmarinifolius, we can see those leaves that are linear. They sort of look like rosemary leaves, but they are not. And then we can see Diplostephium azurium has pretty similar leaf shape, pretty similar morphology, vegetative morphology. The floral morphology is different. We can see the heads here in Diplostephium azurium. They are solitary. The rays are colored, while in Rosmarinifolium, in Linochilus rosmarinifolius, we have usually multiple heads that are smaller with white rays. Similar here. So Linochilus crassifolius from Colombia is very similar to Diplostephium oxapampanum from Peru. They both have this sort of reduced, really thick leaves. In this case, the floral morphology is sort of similar with terminal heads, many flowers there. I believe Oxen Pampanum has more flowers, and also the ray flowers are going to be colored. So very cool to see this case on convergent evolution at the genus level, but we also see this at the species level with species that are very similar to each other. So I'm very familiar with now the Linochilus of Colombia because I did my undergraduate and master thesis in these plants, and I've actually been writing monography, a taxonomy monography for these plants. So I will always be puzzled about the speciation of these plants in the Paramo. So this genus is the third most biodiverse genus in the Paramo. Um, it's found in both the Paramo and the upper limit of the Andean forest. As I mentioned before, we have some Andean forest species that look like small trees, and then we have dispersion by wind. So when I think about speciation in the Paramo, and some of you might be associated or might have read about the Paramo, you know that the Paramo is this high Andean ecosystem, mostly located in Ecuador, Colombia, and Venezuela. It's pretty humid, found about the tree line, high precipitation, and with the island-like distribution. So it happens that Linochilus pretty much follows the distribution of the Paramo, pretty much up to Ecuador. It sort of stops here, although there is one species of Linochilus that is found in Peru. So with this high-resolution phylogeny that I was able to build for my first chapter of my dissertation, I was thinking about testing different modes of speciation in Linochilus. And I was basically trying to contrast two different hypotheses. So one hypothesis would be allopatric isolation or geographical isolation, in which you have, a speciation by geographical isolation. This could happen in two different ways. We could have, a founder event from one island to another, or we can have some sort of vicariance. So I'm not going to be able to differentiate those two in this test, but at least I will know if allopatric or like geography is driving this. So the prediction under this hypothesis is that if geographical isolation is driving speciation, at least recent speciation, we should expect sister species to be allopatric from each other, right? We should not expect sister species to be living in the same island, right? An alternative hypothesis is one of ecological divergence, and this mostly deals with hypothetical local adaptation in which you could have potentially a species. Let's say there is a species A that lives in the forest, and then a population starts to evolve to live outside of the forest, like above the tree line in this area here, right? If there is local adaptation and if there is some sort of genetic isolation over time, we should see that the species that is above the tree line, in this specific case, should develop smaller leaves, should become more shrubby, while species A should retain those sort of ancestral characteristics of being tree and having larger trees. In this specific prediction, what we expect is that sister species might be found in the same Paramo Island, with the difference being some ecological divergent characters, right? So in this case, my ecological divergent character is the leaf area. And we know that there is a clear association between the species in the forest having sort of like regular leaves, like medium leaves, I will say, and then species outside of the forest above the tree line being microphilous, so having really small leaves. So in addition to test for this, so I use the phylogeny that I just talked about. I use herbarium specimens to map the distribution of sister species and see if they were allopatric or sympatric. And then I calculated leaf areas from herbarium specimens to use them as a proxy for ecology. And basically, this is what I'm trying to see. I have two sister species and try to compare, if they live in different islands and if their ecology is different. I acknowledge this is only one character, but it's a proxy for ecology. So I'm going to show you two slides with results. This is the first one. So this is the phylogeny of just Linochilus, which is being calibrated to time. And there is a lot of information here, but what I want you to focus on is this part of the figure. So in this part of the figure, I have plot the box plots of the areas of the leaves. So you can see the leaves that are really small, linear, are gonna be in this area. The box plot is gonna be in this area. This is sort of like also microphyllid leaves, but a little bit larger. And then the largest leaves that we can find in the genus that are gonna look sort of like this. When we look at this box plot, we can see a clear signal in which closely related species definitely tend to have similar leaf areas. So this seems to be a character that is driven by phylogeny. And interestingly here, we can see that all the species that live in the forest that have leaves that are larger, they are part of a single clade that I'm calling here D. So when I look at the phylogeny and then I take out these boxes, which are the sister species, right? And then I do the actual comparison between those sister species. This is the table that I get. So this is one of the main results. So you can see here the comparison between leaf areas between sister species. Notice that only two of those are significantly different here. When we look at the distribution of sister species, we see that most sister species are allopatric. So we can use this to infer divergence. And when we do this, we sort of infer that geographical isolation, it seems like explain 80% of the divergence between sister species. And ecological divergence, oops, I'm sorry, seems to explain only, oh, I'm sorry, this ecological divergence, 13%. And then we have a couple of cases in which we cannot really say with confidence what produced the divergence between sister species. So allopatric speciation seems very important for these plants. That's not surprising seeing the distribution of the plants, right? Seeing the distribution of the plants, but also seeing the distribution of the ecosystems that they are associated with. And so this is a clear example of this. This is sister species, the first one that was in the table, Linochilus phylicoides and lacunosus. They're very similar to each other. They live in different islands. Lacunosus only lives here. Phylicoides is more widespread. And we don't find a significant difference in the leaf area, but they live in different places, indicating that there is a major role of geographic isolation in speciation here. Okay. I think I'm running out of time, so I'm just gonna stop here, and I'm just gonna say that, yeah, geography plays a major role in recent speciation, and ecological divergence events are few, but perhaps they can have a significant impact on greater time scales. So I'm gonna stop here and see if you all have any questions. I wanna thank all the institutions that have helped me, including Herbaria and Botanical Gardens. Thank you.
Great talk, Oscar. Does anyone have any questions for Oscar? So I can do a quick one if someone doesn't have any. So you looked a little bit into hybridization. Is there anything kind of interesting outside of your target group that you found? So like outside, I don't quite remember the names, like the
Yeah,
Dipso
yeah,
and then the
Linochilus and
yeah,
Diplostephium, yeah.
yeah.
Yeah, I mean, I think there is a couple of things. I think first, it was surprising to find recent hybridization among genera. Um, and there is also ancient hybridization among genera, which that's not that weird because at the time when they hybridized, they were probably, closely related. That's one thing. And then It seems to me when you look at the mitochondrial phylogeny, in the mitochondrial phylogeny, actually the large Diplostephium seems to be monophyletic, and I wonder if there is some selection for the mitochondria because of high elevation. That's something that I haven't tested, but I find it interesting. Mhm.
That's great. Thank you. Isaac, if you would like to do your question.
Thanks for the great talk, Oscar. Uh,
Thank you.
I just wanted to ask about this interesting, maybe it's an outlier for the group, but the Diplostephium from the Atacama Desert in Chile that was recently discovered and described. And if
Yes.
had a chance to look at that and have any thoughts about like its weird, outlier
Yeah, no,
status.
that's yes. I reviewed that paper and I was super excited with that paper because it would be really interested to place in the phylogeny and maybe see if it is sister to the rest. There is definitely in Diplostephium sensu stricto. It seems that the most basal taxa are the ones that are in the desert, so in southern Peru. So that's definitely a good observation there, and I'm excited to see where it shows up. Hmm. Yeah.
Mauricio, if you have a quick question, we can do yours and then we can go to the next speaker.
Yeah, quick question. Great talk, Oscar. So you mentioned that you find that hybridization between Diplostephium and Parastraphia.
Hmm.
So there's that species of Diplostephium that occurs in northern Chile, or at least one of the two or three
Hmm.
that currently recognized. But you found hybridization only occurring in that stretch of the genus, or you find indications in some other places?
Well, it's difficult to say. I think I found it in several places, but the test doesn't tell you exactly when hybridization happened. It tells you it happened in those lineages. So, I mean, it seems to be happening all over the place. But at the time, the methods were not strong enough to tell me more information. So, my guess is definitely happened all over the place. And that specific case with Parastraphia, it created a new species that is out there, um, and which is interesting. Yes, and we are describing that with a collaborator in Peru. Yeah.
Okay, thanks you.
Okay.
All right. Thank you so much, Oscar. So now we will go to our second speaker today, who's going to be Glynis Goodman-Cron, who's going to be presenting on the biogeographic history of Helichrysum in the Great Escarpment.
Thanks, Erica. Let me see if I can share my screen and get it to give you start slideshow. Oh dear, from beginning. There we go. Is that it? Working? You've got it.
Yes, we
Yay.
can see it.
Excellent. Thank you very much. And thanks for this opportunity to present on this work. I'd like to acknowledge my co-authors and particularly Nicola Berg. She and I have been working on Helichrysum for more than 10, 11 years. So it's been a great, interesting project. Thank you. Okay, so I love mountains and I love daisies. So this was a sort of natural choice for a topic. And I wanted to look at the role of our Maloti-Drakensberg mountains in the evolution of the Southern African temperate flora. And with Helichrysum being one of the largest genera in that region, it seemed like a natural choice. So just to give you a little bit of background here, Southern Africa is has a very rich temperate flora. And by Southern Africa, I'm referring to the countries that are indicated in the right-hand map there. Oops. They have about 50% of the African plant species richness, according to Peter Linder's 2014 paper, and more than 10% of the world's vascular plants. And the levels of endemisms are also very high. Portions of this flora have more than 70% endemism, which is thought to reflect a long history of in situ radiation. If we have a look at this species richness, can you see my full screen, or am I showing you the participants?
We can, so we can just see your slides.
Okay, great. All right, so this is a map that Peter Linder put together showing species richness in sub-Saharan Africa. And what I want you to notice, obviously we've got the Cape Floristic region down here in purple, with over 500 species per grid square. And then notice that much of the other species riches is along the margin of the subcontinent. So before we get back to that, I just wanted to ask, why such a rich flora? Well, there are a number of hypotheses that have been put forward. One is geographic isolation, and that's certainly true of the Cape Floristic region at the tip of Africa here. Environmental heterogeneity is another, and if you look at this vegetation map that's on the right-hand side below, you'll see that there's quite a complex of different vegetation types in southern Africa. And then Richard Cowling suggested that long-term climate stability might be one of the reasons for this richness of the flora. And then also, Linder and Verboom showed that there's been a lot of cross-seeding of radiations within the sub-region of southern Africa. And once again, you might notice that a lot of this has occurred along the along the margins of the subcontinent. And one of the reasons for this richness along the margins of southern Africa is that that is where the Great Escarpment lies. It's about 5,000 kilometers long, and it was formed after the splitting of Gondwana, and it's thought that it was by passive erosion of that continental margin and parallel retreat that we have these chains of mountains and escarpment. The southern region, so there are three recognized regions. There's western, a southern, and then an eastern one. And right over here is the Maloti-Drakensberg, or the Drakensberg Mountain Center, as it's also been previously called. And the southern section is basically in the Cape Fold Mountains, and it is an important corridor. It's thought to be an important corridor for species moving from the arid west to the much wetter eastern parts, and particularly to the Maloti-Drakensberg region. The Maloti-Drakensberg itself, which you can see illustrated on the right here, this is just a region in the northern part. And that photograph is taken from right on top of the plateau, looking across to a section that sort of curves around. This is called the amphitheater. So you're standing on one side of the amphitheater looking across to the other. So it's essentially a high plateau, but with mountain peaks and a very sharp escarpment on the eastern side. And the highest point is at 3,482 meters above sea level. And that is the highest point in southern Africa. And it's on this plateau and on the mountain tops here that we get alpine conditions, or what are considered here to be alpine conditions. And this is where it is located. So it's very much in Lesotho. The Maloti Mountains are here running through the center of Lesotho, which of course is landlocked within South Africa. And then the Drakensberg escarpment is running around along the eastern border, sort of bordering both of them. So if one climbs up to the summit, you start to enter into Lesotho. But the escarpment itself is more or less in South Africa. And it's part of the Afromontane archipelago of White, 1978. So that would be it there, that little purple spot. And it's thought to be an important stepping stone along the Afro-temperate track. So as I said, we decided to use Helichrysum as the largest genus in the Maloti-Drakensberg. There are about 102 species, many of them endemic or near endemic. And these are just some of them. These 3 lineatum, Milfordia, and we think this is Eclonus. They're not so easy to identify sometimes. These 3 are alpine members, and Helichrysum tenax is more of a shrubby plant which grows in the lower parts of the Drakensberg. And it's got a very sticky leaf, so it's an easy one to identify. So as I said, our aim was to explore the role of the Great Escarpment, and specifically the Maloti-Drakensberg, in diversification of the Southern African temperate flora. And we decided to do this by investigating migration routes of Helichrysum within the region. Oh, and here's this amphitheater at the top. So I was standing about here where there's a large waterfall and looking across to this side here. And then this lower photograph is taken in the central Drakensberg region. So we created a dated phylogeny of Helichrysum based on the same ITS and ETS regions that Goodman-Cron had used previously. We included some outgroup Astereae, members of the Nephalae, and obviously of the Hapclade. We managed to include about 78% of the Helichrysum species of Southern Africa, along with representatives of the various genera that are lodged within Helichrysum, because as I'm sure you know, it's not monophyletic. And we dated this phylogeny using 2 pollen dates for pollen fossils, the first being an ambrosia type pollen, which is illustrated on the right-hand side here, and then a Stevie elytra papus type pollen for the age of Seriphium, and then the mean of 3 studies for the crown age of the Nephalae. We used for our biogeographic analysis, an SDEC analysis, and we coded 9 georegions. We based those on the biomes of Machida and Rutherford, and you can see them illustrated on the right-hand side here with the different colors. And we also included the Afromontane region, and we had to group it with Madagascar, which seemed the best option, just for computational reasons. It just got too large if we put everything separate. And for our distributions, we based it on the literature as well as the South African National Biodiversity Institute's website for distributions of species. So we put forward 3 possible scenarios that we think that Helichrysum and therefore the Hapclade would have migrated northeastwards to the Maloti-Drakensberg via the southern Great Escarpment, as suggested is a common route by Clark et al. In 2012. That's Ralph Clark, who worked in the region a great deal in the southern escarpment region. Or it might have moved via the central plateau, the interior plateau of southern Africa, excuse me, which is at a high elevation, but not nearly as high as the mountains themselves. Or possibly via the Cape Coastal region along here into the KwaZulu-Natal Midlands, which lie just below the Drakensberg region, or possibly just by direct distance, long distance dispersal. So this is our phylogeny. And as you can see, the origin of Helichrysum is, as in other studies, shown to be in the succulent Karoo region, but also in the central plateau to some extent. And we dated its origin to about 25.5 million years ago, so in the late Oligocene to early Miocene. And that is pretty much the same as what has been shown in other studies. Okay, so if we look at the phylogeny itself, you'll notice that there are actually two major clades diverging from the base here. The first of these, clade 1, is relatively small, with clade 2 being much of, well, all of this, the rest of it. Okay, and clade 1 is itself divided into two clades. So we've got 1A and 1B here. And for the most part, this 1A clade is within the succulent Karoo, with dispersals into this core Cape fynbos biome in yellow here. But you'll notice in 1B that there are some green branches, and so that is direct, it appears to be at least, direct dispersals into the Maloti-Drakensberg region with a reverse dispersal back into the core fynbos region. And what's very interesting for me is in clade 1 here, the 3 Drakensberg species that are present there are all alpine endemics. And so I wondered if that was a reflection of their ancestors' adaptation to the succulent Karoo, because at night the succulent Karoo is extremely cold, and the winters in the Cape are very cold, and that's when their rainfall occurs. Drakensberg has a summer rainfall. The rest of the country in South Africa has a summer rainfall. But the Cape can have very, very cold winters. And we kind of wondered if possibly there was almost an element of pre-adaptation from these succulent Karoo ancestors. So I had an honor student in 2020, yes, during COVID. Luckily, we managed to get hold of the herbarium specimens before we all went into lockdown. But Emma Wright looked at some endemic succulent Karoo species and some Drakensberg alpine center endemic species. And this is Helichrysum deserticola and Helichrysum lineatum. And just by looking at them, you can immediately see a whole lot of similarities in terms of the fact that they are very, very small shrublets, so slightly woody, with very small leaves, very compact growth forms. And Emma did a study of the functional traits. So she had looked at a variety of functional traits. These are just the qualitative ones that she looked at. And compared 14 succulent Karoo endemics to 18 of the Drakensberg alpine species. And what you can see is that there are a number of traits that overlap, in other words, are present in both species, in species in both regions. And those include their indumentum, their woodiness, the fact that they have rolled leaf margins, the small leaves, as I mentioned. Also the synflorescent size, the leaves circling capitulum, leaves getting smaller towards the capitulum, and then the fact that they retained their dead leaves on the plants. Succulent Karoo species, however, all had an annual life history, whereas the Drakensberg alpine ones, as one would expect, have a perennial life history, along with these other characteristics. And in this NMDS of these species, based on these functional traits, but in this case, qualitative and quantitative, again, you'll notice that there's quite a large area of overlap in this functional trait space, but then also there are some species that really are much more adjusted or adapted to alpine conditions in the Drakensberg, and there are some that are very well adapted to conditions in the succulent Karoo. Okay, so that was clade 1. If we go to clade 2, you can see immediately that there have been dispersals into many, many places. But I want you to notice at the root of it is a green branch. So it implies that they came via the Drakensberg, the Maloti-Drakensberg. The lighter green is going up into the northeastern escarpment and the Manica highlands of Zimbabwe. The purple is the tropical East African mountains and or Madagascar, and you can see a number of areas where that has happened. The dark blue has not happened very often, and that's the KwaZulu-Natal Midlands. So that's moving down into the lower lands of Southern Africa. And then quite frequently throughout this phylogeny, you can see the lighter green, which is that northern eastern escarpment, and the Manica highlands. And occasionally some yellow, and not very often a reversal back to the succulent Karoo. So here we have some of those regions labeled, which possibly makes it a bit easier for you to see. And okay, so if we look at a summary of the kind of events that happened here, you can see that the analysis suggests that dispersal has been the main mechanism of migration, with much, much less vicariance, and in fact, very little extinction of existing species. Now, that might, of course, be an underestimate. And then if we look at where the dispersals went to, as you might notice, the thickest arrows are here, with about 35 dispersals within our sampling group to the northeastern escarpment and the Manica highlands up here. The next largest number is down into the Midlands, the KwaZulu-Natal Midlands and coastal, and then to this southern escarpment. But what for me is very interesting is that the number of dispersals coming via the southern escarpment to the Maloti-Drakensberg is minuscule. So it suggests that that is not a common route, well, certainly not in Helichrysum. And it seems that the central plateau has played the largest role in providing species. Sorry, that's to it. So the central plateau has played a role, along with the core Cape Fynbos region and the succulent Karoo, from the south northwards. And then we didn't have a large number of tropical or Madagascan species in our data set. So these numbers here are not a good representation of what has happened. Okay. And then what about in situ speciation? Well, we counted about 79 speciation events here. And the arrows indicate endemic genera, so they're occurring only in the Maloti-Drakensberg. But what's quite interesting is that they're scattered throughout the phylogeny, and we only have one rather small clade of endemic species occurring there. So what can we conclude from all of this? Well, the origin is confirmed as the arid western regions of southern Africa in the late Oligocene, with most succulent Karoo species descended from those initial events. And what has not been previously known is that the central summer rainfall plateau has played an important role in dispersal and migration northeastwards. There's been very limited recolonization of very habitats, of those arid habitats, which suggests that the direction of movement is mostly towards the moister, cooler habitats. And the Great Escarpment and the Drakensberg have played a dominant role in diversification of Southern African flora and within Africa. and also that there's been considerable in situ speciation in the Maloti-Drakensberg, but no single large radiation. And so then just a huge thank you to our funders, to Isambelo KwaZulu-Natal Wildlife for the permits to collect specimens and samples, tissue samples. Pretoria University allowed us to take some herbarium tissue samples. A colleague of mine, Jolene Fisher, helped with the map, and my wonderful husband was a frequent field assistant. So, and thank you all for listening and for your interest.
Thank you so much. Does anyone have any questions for Glynis?
I see Jose, I think, has a question.
Jose Mauricio, are you clapping or do you have a question?
Oh, sorry, just clapping. Okay, thank you. Okay.
I would say I can ask a quick question. So, you said that you had around 78% of the South African Helichrysum in your study, correct? The ones that you didn't sample, were those also more endemic species? And how do you think, like briefly, how do you think that that would impact your sampling and your study overall?
Oh gosh, okay. Well, they weren't Drakensberg endemics. I don't think many of them were, but they were obviously hard to get and hard to find because we used, we did a lot of collecting, with Nicola being based in the Cape at that stage, and myself traveling to the Drakensberg quite frequently. And then we were able to access collections at Pretoria University, which Bram van Wyk, if any of you know of him, he's collected extensively across KwaZulu-Natal. So I think we had a good representative sampling. I can't really say if they were endemics or not for certain regions, but very likely. I think it would influence it slightly, but I don't think to a huge degree.
Yeah, I definitely understand that sampling's always very complicated, especially in mountainous regions like that, which are very beautiful as well.
Yeah.
Yeah. All right. Well, thank you so much.